Welcome to the world of mitosis, where one cell becomes two cells that are identical. And I'm going to need your undivided attention because we're talking about mitosis. And here's a joke. Well, not really a joke, a riddle. What divides and multiplies at the same time? Chapter 12, mitosis does. So get ready. We're going in the world of cell cycle and mitosis. Share my screen. Ooh, look at my frozen face. That's upsetting. Okay. And proceed. All right, so we are now talking about, so really quickly, we already went over chapter 16, which was about DNA structure, and DNA replication. And now we're going into chapter 12, which is all about the cell cycle, and in particular, mitosis. All right. Mr. Anderson, I posted this online, I think. The Khan Academy actually does a great job talking about the cell cycle. That would be a really nice place to kind of look with that as well. So what do we need cell division for? And there's four main reasons we need cell division. One, to create new organisms, all right? So remember, for example, if you had a bacterial cell, a prokaryotic cell, what's gonna happen is this thing's gonna get longer, it is gonna duplicate its DNA, and then it is going to split into two. So you went from one cell to two identical cells. Right? One cell to two identical cells. So you literally created a new organism there, right? So sometimes mitosis can create new organism, and that is in particular for prokaryotic cells and also eukaryotic unicellular cells. So organisms that are one cell. Second one is reproduction, right? We need to reproduce, or we need to make more cells, right? The third reason is that you need to grow and develop. So for example, when you were first fertilized, you weren't fertilized, when your mom had an egg and your dad had a sperm and these came together, you now had kind of a fertilized egg, right? We all started with a fertilized egg. That fertilized egg went through many, many, many billions of rounds of mitosis to create what we are today, right? Which is kind of amazing. We're literally growing and dividing these cells to create something very complex. Also in terms of development, right? So we're not only just a sack of cells, we have developed neurons and bones and hair and all this cool stuff. So not only have we grown from that fertilized cell, but also many of our tissues have developed as well. And then the fourth one is for renew and repair. So I'm trying to, I don't have any major injuries right now, which is amazing. But somebody probably will look down their hands and see like a cut or something, right? Anytime you get a cut or an injury, your body is going to try to repair that, clean up the wound, figure that out. And that is all going to be done through mitosis, all right? So really the main part of mitosis is, terrible handwriting, is that mitosis, mitosis ensures that the continuity of life is there. Not good English today. So the continuity of life absolutely depends on cell division, cell reproduction, AKA mitosis. All right, so there are some really cool movies of mitosis. I will post these online. Try to remind me in class and we can watch some of them in class as well. It shows bacterial growth in division, eukaryotic cell division, development from a single cell in a fish egg all the way to a hatching fish, and wound healing. All right, so here's some important terminology that you must know. All right, so let's do a little matching game. All right, so we've already talked about chromosome down here, so let's start with that. So what is a chromosome? Remember that in our previous slides, we talked about chromosome being DNA plus proteins. All right, so I would put that with a very long DNA molecule associated with proteins. All right, genome, sorry, I'm kind of bouncing around, but that's okay. Genome is a cell's entire collection of DNA. All of the DNA in an organism, or all of the DNA within a cell, is called the genome. That's an entire collection of cells, right? Gametes, well, actually, I'm going to start with somatic cells. Sorry, again, bouncing around, don't hate. Somatic cells are all of the body's cells except the reproductive cells, sometimes called germline cells. I often call them our sex cells, right? So all of the cells except for those, right? Meaning that the gametes are the reproductive cells. So the gametes are the germ cells, otherwise called the sex cells, right? In the gametes all have half the amount of DNA, whereas somatic cells have all of the DNA, right? And that makes sense because when we have an egg and a sperm that are going to come together, they both better have half so that when they come together, they're going to have the full set of DNA, all right? So gametes have half the amount, meaning that they are haploid, half the amount of DNA, right? Whereas somatic cells have all the DNA, so they are diploid, right? They have 2 copies of all of the DNA, half from mom, and we're going to see half from daddy. All right. And then last but not least, by a process of elimination, we are dealing with chromatin, right? And I'm just putting out this word because it's something that you might come across. Chromatin is the complex of DNA and proteins, right, that make up the eukaryotic chromosomes, right? So sometimes people will refer to the DNA in the cell as chromatin, but each individual chromosome is a single piece of DNA associated with protein. But chromatin kind of refers to all of the DNA and proteins within a cell, right? Okay, so there are 2 main types of cell division that we're going to be talking about. Chapter 12 is focusing on mitosis, all right? So when we are dealing with mitosis, what we have is the following. We have cells that replicate their DNA, right? So they go through DNA replication, and then they divide. So DNA replication followed by cell division, all right? And this results in 2 identical cells. So notice this here is a diploid cell. That means you have 2 copies of each chromosome, one from mom and one from dad, one from the mom egg, one copy of every chromosome from mom egg, one copy of every chromosome from dad's sperm. They came together to create this diploid cell, which created all the cells in our body through mitosis. So diploid, 2 copies of each chromosome. When they divide, you're going to get 2 identical cells, each with the full copy of 23 pairs of chromosome to give you 46. All right. So the human genome is 23 pairs, right? for a total of 46 individual chromosomes, 23 from mom and 23 from dad. All right. So now the other type of cell division is called meiosis. Meiosis only makes gametes, right? What are the gametes? Those are the sex cells. Only makes the sex cells. It's a very complicated process. And the only reason we do it is to make egg or sperm. That's it. All the other types of cell division in our body are mitosis. Meiosis is only to make egg and sperm. That's it. All right. So cells are going to do the same thing. We're going to see DNA replication, right? We are going to see cell division. They divide into 2 cells. And then we are going to see they divide again to create 4 haploid cells. Notice these are not identical to the diploid mother cell, right? They are now halved to create haploid cells, right? So we're going to really focus on mitosis, but first let's talk about chromosome terminology. All right. So chromosomes by definition are a piece of DNA, one piece of DNA, right, with its associated proteins. Remember, those proteins are called histones. DNA wraps around those histones. And that's how it all fits into a teeny tiny cell that we can't even see. 6 feet of DNA into a teeny tiny cell, right? So cells must replicate their DNA before the cell divides, okay? When those chromosomes are replicated, so these replicated chromosomes are called sister chromatids, all right? Sister chromatids, and these are attached by proteins at the centromere of the chromosomes. That's what holds them together. So let's focus on these kind of cartoon ones first. So here we have a single chromosome, right? It's a single piece of DNA wrapped around histones. When it duplicates, and again, this duplication happens in S phase of interphase. And when it duplicates, you're going to get 2 identical chromosomes, and they're held together at this middle part, and that is the centromere, right? The centromere is the center of these chromosomes. Now we have sister chromatids, right? So we went from a single chromosome to sister chromatids. Those are replicated chromosomes, right? And then when they separate, you are going to go, the sisters, what we're going to see is in mitosis, these guys are going to separate. So we're back to kind of having a cell with 2 identical cells that look just like their mothers. With the mother cell. And remember, these sisters still have a centromere, right? So if we look at this big bad chromosome here, we see that there was 1 chromosome, and then it duplicated to create a replicated chromosome, which are now called the sister chromatids, held together by their centromere. Okay, so mitosis versus cytokinesis. People often use these interchangeably, and that's not actually correct. Mitosis really refers to the nuclear division, right? Inside of the nucleus, in particular, kind of the DNA, right? So mitosis is the nuclear division of a cell after DNA replication, resulting in 2 identical genomes. And we're going to see that this has 5 stages that you're going to know and love. All right. So what we're seeing here, mitosis, is where we have those replicated sister chromatids getting pulled apart into their separate cells. Cytokinesis here is the division of the cytoplasm, right? So this is cytoplasm division to actually form the two separate cells. And this occurs right after mitosis or meiosis, right? So mitosis, nuclear division; cytokinesis, cytoplasm division. All right, so the cell cycle. I'm gonna move my face so you can see a little bit better. Okay, so for the cell cycle, we've talked about this very briefly before, but there are multiple phases of a cell's life, right? The first part is called G1, all right? That stands for either a gap phase. I also call it a growth phase, right? So G1, the cell is going to grow and get ready for DNA synthesis. The S phase is going to be DNA synthesis. That's why it's called S phase, DNA synthesis. Then we go into another phase called G2, which is again called a gap or growth phase, right? G1 is really prepare cell for DNA replication, right? G2 is really prepare the cell for mitosis, right? So it's kind of preparing, getting ready, giving the cell time to get ready for that. All together, G1 to S to G2 is called interphase, right? Interphase. This is where the cell grows and the DNA gets replicated and the cell gets ready for mitosis. 90% of a cell's life is in interphase, all right? Mitosis gets all the business because it's all jazzy and awesome, but really 90% of a cell's life is in interphase, right? Then a very small percent of the cell's life, about 10%, is in M phase or the mitotic phase, right? And that is represented here by these kind of, I don't know, yellowish color. And you have mitosis followed by cytokinesis, all right? And that together is the M phase. And we're gonna see that there are 5 stages that you have to know for mitosis: prophase, prometaphase, metaphase, anaphase, telophase, right? 1, 2, 3, 4, 5. So those are the 5 stages of mitosis. Then this is all going to be followed by cytokinesis, where we have cytoplasm splitting. All right. But again, before you get into this jazzy mitosis phase, remember a cell goes through G1, S phase, where DNA gets replicated, which we've already talked about through DNA polymerase, followed by G2, another growth phase. So let's talk about the phases of mitosis. That's okay. So it is so prophase, prometaphase, metaphase, anaphase, telophase. In regular biology, they don't talk about this prometaphase, but I think it's important. So many people probably learn it as PMAT. Maybe in class, I'll tell you about a really inappropriate way to remember that. All right, interphase. So interphase is the very first part. This is G1, S, G2. Here, the nuclear envelope is still intact. DNA is going to be replicated in S phase. And it's really important that individual chromosomes cannot be distinguished in interphase. It literally looks like, if you've ever taken the leather off of a baseball, it looks like a ball of yarn. That's what the chromosomes look like throughout interphase. G1, S, G2, even during DNA replication, you can't distinguish individual chromosomes from one another. So that is a good indication that we're in interphase. So for each of these, we're going to look at a real picture of a cell and kind of a drawing of a cell, all right? One thing that you'll notice is that in S phase, not only are we getting DNA replication, but we're also replicating I spelled that wrong, the centrosome. Remember, the centrosome has two centrioles in kind of this amorphous blob that is required to grow out microtubules, which we're going to see that are become really, really important. So S phase, you get DNA replication. You're also replicating your centrosomes. That was my whole PhD, was centrosome replication. Pretty fun. Okay, second phase, prophase. All right. So now we're really into mitosis. All right. This is the first phase of mitosis. It's called prophase. And for each of these phases, you should be able to recognize it under a microscope and be able to say what's going on in that cell. So here, this is the first time we see visible chromosomes. So if you see individual chromosomes, and it's a little hard to see in this picture, but you're starting to see individual chromosomes. Remember, those chromosomes are in the form of sister chromatids because they've been duplicated in S phase of interphase. The microtubule, the mitotic spindle, which is representing here, which I'm these little lines, these are microtubules growing out of the centrosome. Those are the mitotic spindle. They start to form. So the centrosomes start to grow out their microtubules. The centrosomes start moving to opposite ends of the cell. And what we see is the nucleolus disappears. And just a reminder, the nucleolus is where we are making our RNA in the nucleus, and it disappears. The next phase of mitosis is pro-metaphase. I will never ask you to be able to recognize prophase versus pro-metaphase on a microscope slide or in a picture. However, you should know that they're different and they have different things going on. So first off, the nuclear envelope breaks down, all right? So that nuclear envelope literally gets broken apart, gets broken down. The spindles invade the nuclear space. So you see these microtubules reaching for one another from opposite sides of a cell. And we get the microtubules, these spindle microtubules, attaching at the middle of the centrosome, at the middle of the chromosomes, called the kinetochore. All right, and I'm going to draw that out in a right here. Okay, so single chromosome. During S phase, we are going to see that that single chromosome gets duplicated. It is held together by the centromere. The centromere is a sequence of DNA, right? The kinetochore is going to be built around the centromere. This is the kinetochore. There's a lot of weird terminology in this section. The kinetochore is protein that's built on the centromere that will bind, I'm going to put spindle microtubules, will bind to the spindle microtubules, right? So the centromere is just a sequence of DNA. The kinetochore are proteins that associate with that DNA where the microtubules are going to attach. All right, metaphase. So again, we're talking, we're talking about mitosis. We've talked about prophase. We've talked about pro-metaphase. And now we're talking about metaphase. This is one that makes sense to students. The metaphase, the way to remember it is that chromosomes line up on the midline. Metaphase, midline. All right. Here we have centrosomes at the fully opposite sides of the cell. and we have spindle microtubules growing out. They're already attached to the kinetochore of these chromosomes. And they have basically a tug of war. They push and pull from both sides until the chromosomes are lined up perfectly at the midline. This is sometimes called the metaphase plate, all right? So if you're ever drawing a cell and you have centrosomes on this side, the midline is going to be between them. If I had a cell and I drew my centrosomes on this side, the midline would be this way, all right? And you just have to make sure that you draw your chromosomes correctly, because there will be some drawing on the test, right? So if you had a chromosome, it is going to line up along the midline, right? So that the kinetochore has access. So on this one, the chromosomes would line up here to give you access to those kinetochores. So the metaphase plate is this kind of invisible plane midway between those two poles where chromosomes are going to line up. Anaphase is the next phase, and this is where sister chromatids get separated from one another, right? So we're talking about anaphase. Here, those duplicated chromosomes get pulled apart by those microtubules, right? Getting pulled apart by those microtubules on their kinetochores. And each of these now kind of chromatids is goes back to being a single chromosome, right? So we're back to a single chromosome. Each pole has should now have identical and complete genomes. So in anaphase, if we draw a circle around both, they should be identical. Identical. All right. Last phase of mitosis is called telophase. And telophase is where you get the spindle breaking down, so those microtubules go away. The nuclear envelope reforms, right? So you can see that happening. And the chromosomes start to become a little less tightly condensed. So more stringy, less tight. And the nucleolus reforms. And that signifies the end of mitosis. Cytokinesis, however, is often clumped in with mitosis. So it follows mitosis. And this is where you get the division of the cytoplasm, and you literally get the formation of two daughter cells here, right? In animals, this kind of structure where you get the two cells kind of pinching in animal cells, that's called a cleavage furrow. And the way that works is actually through microfilaments or actin, right? That's how that kind of cinches together. In plant cells, you have what is called a cell plate. So into plant cells that have those cell wall with cellulose, the cell plate forms, and that is the cell wall forming as well. And that's the end of division, right? So just a picture of what this looks like in animal cells. You get that kind of, I don't know, looks like a butt or peach, that's a better word. And it's called a cleavage furrow. And I always think, I'm like, some dude named that for sure. Anyway, so that's the cleavage furrow. In plants, what you see is this cell plate starting to form. And that is usually formed by vesicles rather than just kind of a contraction of the cytoskeleton. And just so you know, because it's one of my favorite words, the cell plate in plants is also called a phragmoplast. Awesome little Jeopardy word for you. So you should be able to recognize pictures. Sorry, here, I'll put it over here. So you should be able to recognize pictures under a microscope, what phase they are. You should be able to recognize drawings, what phase they are. In both animals and in plants. So again, in prophase, you are looking for the presence of individual chromosomes. You start to see those here. Prometaphase, I would never ask you about that one, so don't worry about that. Metaphase, they're going to line up on the midline. Anaphase, they are being pulled apart. Those sister chromatids are being pulled apart by this mitotic spindle. And in telophase, you start to see kind of the nuclear envelope reforming. All right. So let's do a little test. So A, what do you think this A cell is? Right. So notice, can you see individual chromosomes? Yeah. But are they lined up in any way? No. So I would call that prophase. Notice most of the cells, I'm drawing a bunch here, most of these cells are, that's actually 2 cells, most of these cells, you can't see individual chromosomes. Those are all in interphase. So C over here, that would be interphase, the majority of cells. Remember, 90% of a cell's life is in interphase. B here, we are seeing that those sister chromatids are being pulled apart. So this would be anaphase, right? Just to give you a good sense. So this I would call prophase, 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 and I don't see anything else. Okay. That was animal cells. So in plant cells, so let's look at A. So here we can see that they are spreading apart or being pulled apart. So that one is anaphase. B, this looks like the majority of the cells, right? There's lots of other cells around here that look just like B. Can't really see individual chromosomes. So I would call that interphase. C, this is a cell that was just divided, but you can see new nuclear envelopes forming. So that would be telophase. And D, here's the cell. You can see individual chromosomes, but you can kind of see a line here, right? The metaphase plate. So that would be metaphase, all right? Something like, let's see, something like this is probably prophase. You're starting to see individual chromosomes lining up. This cell here, I would say that's kind of a toss up between maybe metaphase versus anaphase. You can't really tell. I would not give you something that is in between. I would give you something that you should really be able to hammer down. All right. So we will stop there. That gives you hopefully a sense of mitosis, and we'll finish this next one in another recording. Peace, and I'm out. Bye. Adios. Ciao.